All right, guys. Good morning. It's my pleasure to be here to share my recent research especially we are interested about like a single cell data analysis, like the deep learning. But this topic is pretty little bit old, but we take for example a long time. So we are working on one specific study about like what's the next negative negative binomial distribution related. Okay, we will talk about like two things. One is about like theoretical framework of the difference of two negative binomials. And the second is how apply this theoretical result to study to do the significance testing for like a single cell RNA-seq. Actually, it can be broadly applied for different of the data set, especially for the dispersed count data. Okay. All right, so let's go very quickly. So like the sequencing technique definitely is quite fundamental and essential recently in the last 20 years. And the RNA-seq and the single-cell RNA-seq is most important. And so simultaneously, like chromatin detection, this is years ago we work on like capture methods to detect the chromatin detections. We all have such questions, reason about like how can we compare to samples or populations to detect the significance between them, like for one gene. Okay, so like all one peak region. For example, here, like there's chip-seq data. Like you have different peaks across the different of the samples. So like how you compare them. Yeah. If we go back to very basic one, so we may like ask, okay, you have PCR replicates. What do you want to do? Compare the significant difference between them. What do you want to do? Everybody will first try student t-test. Yeah. That's based on normal distribution hypothesis. Right? If you have two populations like following negative binomial, no, I'm sorry, normal distribution, then so you want to compare the mean of difference of them. So you want to do, okay, so student t-test. If you don't know what's the distribution of your two data sets, what do you will do? Yeah, Wisconsin is the non-parametric methods. Yeah, those methods it works. However, the non-parametric, parametric will have lost statistical power. We will go back to this point later. Okay, so like, how can we take this like a single cell RNA-seq data, for example, how can we compare these two? For each of this figure I show you, like for this one row is one gene and the column is the different cells. And you have the, for example, I have two conditions. Okay, two experiments, for example, cancer, non-cancer, and those cases. So you can see, okay, so there's genes, there seems like show this significant difference. Yeah, some of them maybe not. Yeah. So like definitely like we have methods. We have DSeq2, we have a HR. Uh, Even for chip-seq, we also have some of other like methods like yeah, max2. Max2 as very first version used the Poisson distribution to model the peak depths. And then so also use the negative binomial for this, to mimic this the distributions. Okay. All right, so there's the question. So if you have two of these like populations, you have the data, the samples, you want to compare them. Yeah. So if there's the vastly very popular distribution so far is negative binomial. There's a two-parameter model, so it can be used because usually we observed in counting data, especially for sequencing data, it follows the heaven tail distribution. the heaven-tail distributions that means like it's non-symmetric. It has somehow like a very long heaven-tail. They are like typically the variance is bigger than mean. So in that case, negative binomial is usually the first choice. All right, so how can we do that? Yeah. So we use negative binomial to simulate this, but how can we compare them like to find their significance? So let's go back to a little bit, go to the negative binomial. This is a quite old distribution, almost 400 years ago. So Pascal formatted this distribution. And so you can see here, so just like some of the heaven tail, especially for the R, smaller R. And that means, yeah, so like the R and the P mainly, usually you can consider like the R is the failure and the P is the failure probability. Then, so you can use for any of those the variable, you can think of like the success of your sequence rates. Okay, and so how many of those the key success rates can be detected if following there's R and P, there's the negative binomial distribution. Okay, so theoretically, in the last 100 years, this distribution well studied, and many of these generations had been established for like convex combination of a set of this negative binomial. But the difference of them haven't been set up yet. 10 years ago, so we want to start it like what's the difference? 10 years ago, like there's a paper talk about this issue. At a very special case, if the R1 equals R2, they give the result. However, this result can't be applied directly for our like the comparison, a comparative analysis for our like single cell RNA-seq or like bulk RNA-seq, because you suppose R1 equals to R2, it doesn't make sense because ours like research like experiments come from two independent experiments. So it's reasonable to assume they are independent samples. Okay. All right. So then, so like that's our result. So we actually years ago we started to for this analysis for at that time I worked at UT Dallas and collaborated with the professors from like UT Southwestern. So we study the chromatin interactions. So they asked, okay, can we do some of the testing for this? I checked, okay, so there seems like no theoretical result for the difference of two negative binomials. So I start to derive those formula. And so there's a little bit difficult for this one because negative binomial is defined under non-negative integers, but the difference of them actually defined among the whole integers, positive or negative. So, but anyway, so finally, so we get the result like the PMF and the CDF and the mean variance. So we set up this, the formula. The good thing is if we have those, formula, those result available, then so you needn't depends on like empirical distribution, MCMC simulations to get these calculations. So you can directly use the formula to calculate what exactly they are, okay, for the testing. Anyway, so I just ignore the details like for those proof. So we also consider a little bit like this asymptotic properties about this distribution. So like from the positive and negative depends on which distribution may dominate the final, like the whole property. Yeah. Okay, so this is the theoretical result. Let's quickly have like a simulation to check like what it looks like. So the first figure I show you like for 100,000 simulations the samples of this two negative binomial. This is one negative binomial, another negative binomial, and this is the difference of them. Yeah. If we compare the difference, calculate the pair difference, and calculate the frequency, so you pretty like that. So the red one actually is our theoretical distribution calculated directly from our formula. Okay. And so here there's another combination. So if also the same numbers of samples, so you can see, yeah, so there's this, sorry. Yeah, there's the one negative binomial, another negative binomial. If you difference, it looks like that. Yeah. And the red line is the theoretical distribution. We call this dot MB. Here I use the C and D to show you if you reduce the simple numbers to 10,000 and then further to 2,000, you can see if you trying to mimic this, the distribution, negative binomial distribution, you will see the variance, okay, significantly increased. Okay, if you can consider like from the real study, the noise increased dramatically. So in that case, if your calculation, the comparative analysis depends on empirical estimation of those distribution, you may cause to limited statistics power, okay, especially for small sample numbers, especially like for the real cell types. You may have just tens of these cells only, not hundreds, not thousands. Okay. And so, okay, so with this theoretical result established within, so applying this to single cell data analysis, we're trying to see, okay, can we use this theoretical result to improve the differential, the comparative analysis for two groups of this single cell types, and to detect the differentially expressed genes. Okay. Then, so the idea here is straightforward. The key is use our distribution, so to calculate the p-value, calculate the p-value, and then so we use some of our permutation-based methods to establish the PDF, to also like to resolve those so-called the imbalanced data set problem, because usually when you compare two of them, so you can guarantee there's two data sets, they have same numbers of cells. Okay. All right, so let's see what is the performance. We compared with like the DSeq to like HR, and this is several popular methods. You can see, so like overall ours AUC and is better than them. Even not that significant, I would say, like it's not that many big pro improve, but this is based on large numbers, not large cell numbers. That means if you have thousands of cells, yeah, all these methods work well, because you can, based on this large number, you can well estimate even the empirical distribution. It's no problem. Yeah. All right. So the B we're showing like if there's some of dropouts, what will happen? So here we artificially introduce like the dropouts levels, and you can see, so with the increased dropouts rates, our the performance definitely decrease, but we keep the top. Okay, keep it top. And absolute value is still reasonable. Yeah. Okay. And so this C we showing like, since we have the closed forms to use to calculate this, the difference, the probability. So we suppose we can get fast runtime. Okay. So then, so there's it here. So it's here is overlapped with another one also based on empirical based methods, pretty similar. Yeah. Okay. And the first one we test Okay, so how the simple numbers will affect the sensitivity or the performance. Okay. So like with the 20, 50, and 2000, 2500 cells, we can see, so this is the sensitivity numbers. And when, especially we observed for small cell numbers, actually we achieved the super sensitivity for this case. To further check this point, so we further like checked for small cell numbers, okay, and for this case we also simultaneously checked like the imbalanced data set. Yeah, so like you can see here, like the 100 over 100 and like 100 over 75. Yeah, there's imbalanced case. So for the imbalanced data set, so if you have one or two, so you want to have the difference of them. So to calculate the difference, this actually is based on, you want to calculate the simple mean of this two difference. Okay. Then, so that case, so we have two strategies to calculate. One is usually like the DSEQ2 HR use the subsampling. The subsampling means if you have a large data set, a small one, so you just randomly sample a small site to with the same size of the small one, calculate the difference. Okay. Another strategy is random assignment. You can also like keep the large one. For the small one, you randomly select and compare them. Okay. The big one. You want to use all the cells for the from the large data set. Okay, so these two, actually from our comparison, we're showing they're showing pretty similar, the performance. And only for the sensitivity point of view, there's one seems showing significance, like based on our turnarounds are about like the imbalanced for this subsampling seems even better and better for this case, for the sensitivity. So in our methods in the pipeline, by default we use the subsampling. Okay, and we also compare with HR and DSEQ2 and the Wisconsin Coxin methods. So you can see overall ours F1 is bigger, higher than this, and overall like HR seems to show better than the DSEQ2. Okay. And so, but when this one only at this last combination, like 25 by 25, it's a big drop. Yeah, big drop. So this one is the non-parametric measures. So you will see the overall the performance is not that good as the parameter-based measures. Okay. Okay, we applied these measures to two case study. One is for the prostate cancer, and then another for another for this is for mouse memory study. We check like what is the difference of this neurons with and without memory, what's the significant change in the genes. And then so we find like our methods, like you can see our methods get more sensitivity. Actually, we detect most, the genes, even they have a small photo change. The x-axis is photo change. So this DSEQ2 result from their author's original analysis, you will see our probability, the p-values, even for there's some of a small photo change, you still can get some significance. It's not just like this DSEQ2's result. Okay. All right. So we implemented this .nb for 5 languages. You encourage you to use it. Yeah. If later so you want to compare two of them, like not normal distributed or like not something, you have a new methods now. Okay. You have a new text statistic tools can use. Okay. All right. So for summary, so we give us some of theoretical result, hopefully we make up the missing piece in this like a discrete distribution. And also we, by applying these methods, the model, the theoretical result, we design a new methods for differentially the comparative analysis. So we see it's super performance. We have just quickly, so we have a PhD position open in my lab. So we work on this, the the single cell generative AI and those stochastic optimization and control related topic. So, all right. So finally, I will thank our lab member and also there's grants from ISF and RH. Okay. Any questions? Yes.
Have you considered theoretical implementations of the zero-inflated negative binomial distribution?
Very good question. So this is next step. Actually, our paper.
Could you repeat the question?
I'm sorry?
Could you repeat the question for the virtual?
Okay, yeah. So the theoretical, I think the question is how consider the theoretical difference of the two zero-inflated negative binomial. Yeah, zero-inflated negative binomial definitely also applied in the single cell data analysis because of the dropout question. So we are considering this because if you have this negative binomial, you're theoretically the two VI and B distributions, you only have a difference of the zero, like a constant zero part, okay, the term, and plus another dot NB, yeah. So we can estimate these two, yeah. So the constant zero part is easy to resolve later. Okay, but anyway, so there's I think that's a very good question.